Hello everyone, welcome back to Blooming Botany and Bindas Botany. Today I am going to discuss the most important MCQ on reproduction in flowering plants, that is development of embryo in monocot and dicot. This MCQ will help those who have prepared for NEET biology, TGT biology, PGT biology, common PG entrance test, common university entrance test and other competitive examination. Let's begin. Those who have new to my channel, please subscribe my channel and press the bell icon for notification. Question number 1. The development of a dicot embryo is called option A monocotyledonous type, option B crucifer type, option C caryophyllaceous type, option D asteroid type. The correct answer is option B crucifer type. The next question number 2. First scientist to study development of angiosperm embryo was option A. Statsberger, option B. Fleming, option C. Hanstein, option D. Hofmeister. Correct answer is option D. Hofmeister. Hofmeister was actually the first scientist to describe embryo development in angiosperm. The next question number 3. Embryogeny in capsula was first of all studied by option A. Hanstein, option B. Sauges, option C. Hamilton, option D. All of the above. Correct answer is option A. Hanstein. The development of a dicot embryo, specifically the crucifer type or onagrad type, was first studied in Capsula bursa pastoris or Shepherd's Purse by Hanstein in 1870. The next question number 4. The dicot embryo development from option A zygote, option B antipodal cell, option C endosperm, option D egg apparatus. Correct answer is option A zygote. The next question number 5. In dicot embryo, the first division of zygote is option A vertical, option B transverse, option C diagonal, option D random. Correct answer is option B transverse. The next question number 6. The first division of zygote in dicot result in option A 2 equal cell, option B 2 unequal cell, option C 4 cell, option D suspensor. Correct answer is option B 2 unequal cells. The next question number 7. The smaller terminal cell of zygote form option A suspensor, option B embryo proper, option C endosperm, option D seed coat. Correct answer is option B embryo proper. The next question number 8 is the larger basal cell of zygote form option A embryo proper, option B suspensor, option C cotyledon, option D root apex. Correct answer is option B suspensor. This is the zygote. The zygote divided transversely to form the upper small terminal cell and the lower large basal cell. The terminal cell further divided to form embryo proper and the basal cell divided transversely to form the suspensor. The next question number 9. The first recognizable stage of dicot embryo is option A heart-shaped, option B globular, option C torpedo, option D mature. Correct answer is option B globular. This is the globular embryo. First, the zygote divide transversely to form the terminal cell and the basal cell, and it repeatedly divide to form the globular embryo. Then it form the heart-shaped embryo. Then it form the torpedo embryo. Then it mature. And this is the suspensor. The next question number 10. The function of suspensor is option A to provide water, option B to provide oxygen, option C to push the embryo towards endosperm to provide more food, option D to suck food. Correct answer is option C to push the embryo towards endosperm to provide more food. The next question number 11. The basal cell of suspensor functions as option A hypophysis, option B haustorium, option C embryo proper, option D cotyledon. Correct answer is option B haustorium. The next question number 12. The uppermost cell of suspensor is called option A hypophysis, option B apical cell, option C terminal cell, option D embryo cell. Correct answer is option A hypophysis. This is the suspensor cells. The uppermost cell of suspensor is called hypophysis. This is the uppermost cell. It is called hypophysis. And the basal cell of suspensor functions as a haustorium. This is the basal cell of suspensor and which functions as a haustorium. The next question number 13. Dicot embryo consists of option A radical and plumule, option B radical, plumule, cotyledon and sometimes endosperm, option C radical, plumule, cotyledon and tegmen, option D radical, plumule, cotyledon, tegmen and testa. Correct answer is option B radical, plumule, cotyledon and sometimes endosperm. The next question number 14. In dicot embryo, hypophysis is option A. Terminal cell of suspensor that grows to push the embryo downward. Option B. Middle cell of suspensor that grows to push the embryo downward. Option C. Terminal cell of the suspensor that form radical. Option D. Part of embryo that gives rise to radical. Correct answer is option C. Terminal cell or lowermost cell of the suspensor that form radical. This is the hypophysis which form the root tip or radical. The next question number 15. A typical dicotyledonous embryo consists of option A radical only, option B embryonal axis and cotyledon, option C cotyledon only, option D embryonal axis only. Correct answer is option B embryonal axis and cotyledon. The next question number 16. The correct sequence of embryogeny, that is development of embryo in dicot is option A zygote, then globular, then heart-shaped, then torpedo, then mature embryo. Option B zygote, then form heart-shaped, then form globular shape, then form torpedo, then mature embryo. Option C zygote, then torpedo, then globular, then heart-shaped, then mature embryo. Option D zygote, then torpedo, then heart-shaped, then globular, then mature embryo. Correct answer is option A. First the zygote form, then form globular embryo, then form heart-shaped embryo, then form torpedo embryo, then it mature. The next question number 17. In dicot embryo, the radical is formed by option A epibasal tire of embryo, option B hypobasal tire of embryo, option C hypophysis of suspensor, option D terminal cell of suspensor. Correct answer is option C hypophysis of suspensor. Radical is formed from hypophysis of a suspensor. This is the hypophysis structure where the radical is formed. And this is the basal cell. It is also known as ostoium. The next question number 18. The part of the embryo between the cotyledon and the embryonic root, that is radical, is called option A hypocotyle, option B epicotyle, option C plumule, option D coleorrhea. Correct answer is option A hypocotyle. This is the cotyledon and this is the radical, and between the part is called hypocotyle. This is the hypocotyle, this is the cotyledon, and this is the radical or root part, and between the part is a hypocotyle. The next question number 19. The part of the embryonal axis above the level of cotyledon is called, or the portion of embryonal axis above cotyledon is called as option A hypocotyle, option B haustorium, option C hypophysis, option D epicotyle. Correct answer is option D epicotyle. This is the cotyledon. This is the epicotyle and this is the hypocotyle. This is the embryonal axis. Above the level of cotyledon is called epicotyle and below the level of cotyledon is called hypocotyle. The next question number 20. The portion of the embryonal axis between plumule, that is future shoot, and the cotyledon is called option A hypocotyle, option B epicotyle, option C coleorrhea, option D coleoptile. Correct answer is option B, epicotyle. This is the epicotyle part. The next question number 21. Hypophysis gives rise to option A, root cap and root apex, option B, cotyledon, option C, plumule, option D, endosperm. Correct answer is option A, root cap and root apex. This is the plumule part which form the shoot, and this is the hypocotyle, and upper is the epicotyle part, and below this it is the radical which form the root, and these are the cotyledons. Next question number 22. Cotyledons are option A, modified roots, option B, embryonic leaves, option C, known as primary endosperm nucleus, option D, present in gymnospermic seeds only. Correct answer is option B, embryonic leaves. The next question number 23. Cotyledons and testa respectively are edible part in option A, French bean and coconut, option B, cashew nut and lychee, option C, groundnut and pomegranate, option D, walnut and tamarind. Correct answer is option C, groundnut and pomegranate. Cotyledon is the edible part of groundnut and testa is the edible part of pomegranate. The next question number 24. Embryonal axis of seed is called option A, tegellum, option B, scutellum, option C, codon, option D, cortex. Correct answer is option A, tegellum. The next question number 25. Study of seed is called option A, embryology, option B, palynology, option C, spermology, option D, pomology. Correct answer is option C, spermology. The next question number 26. The part of the embryonal axis between plumule and cotyledonary node is called option A, epicotyle, option B, hypocotyle, option C, hilum, option D, raphe. Correct answer is option A, epicotyle. The next question number 27. The part of embryo axis between radical and cotyledonary node is called option A, epicotyle, option B, hypocotyle, option C, hilum, option D, raphe. Correct answer is option B, hypocotyle. The next question number 25. Plumule and cotyledons are developed from which part of the embryo? Option A, hypobasal cell, option B, epibasal cell, option C, hypophysis, option D, suspensor. Correct answer is option B, epibasal cell. This is the epibasal cell and lower is the hypobasal cells. The next question number 29. From which of the cell of embryo plumule is produced? Option A, apical octant, option B, proembryo, option C, hypophysis, option D, micropylar embryo. Correct answer is option A, apical octant. The next question number 30. From which cell peripheral region of radical is produced? Option A, vegetative cell, option B, hypophysis, option C, apical octant, option D, micropylar octant. Correct answer is option B, hypophysis. This is the hypophysis cell which form the radical. And this is the swollen vascular cell. It is called hausteria. The next question number 31. In flowering plant, hausterial cell is option A, the cell of suspensor present at the micropylar end and formed during embryogenesis. Option B, the cell of suspensor present at the chalazal end and formed during embryogenesis. Option C, the cell of suspensor present at the micropylar end and formed during the formation of the endosperm. Option D, the cell of suspensor present at the chalazal end and formed during the formation of endosperm. Correct answer is option A, the cell of suspensor present at the micropylar end and formed during embryogenesis. The next question number 32. The first cell of suspensor towards micropylar end is called option A, vesicular cell, option B, hausterial cell, option C, hypophysis, option D, both and B. Correct answer is option D, both and B. Hausterial cell is also called vesicular cells. The next question number 33. Role of suspensor is option A, to push the embryo in the nutritive zone, option B, to derive nutrition from the embryo, Option C, to help in the fertilization. Option D, to help in maturation of the embryo. Correct answer is option A, to push the embryo in the nutritive zone, that is towards endosperm. The next question number 34. The function of suspensor is option A, to provide water, option B, to provide oxygen, option C, to push the embryo towards endosperm to provide more food, option D, to suck food. Correct answer is option C, to push the embryo towards endosperm to provide more food. The next question number 35. The length of the suspensor is option A, 1-celled, option B, 2-celled, option C, 3-celled, option D, 6 to 10-celled. Correct answer is option D, 6 to 10-celled. The next question number 36. Heart-shaped stage appear due to the formation of option A, cotyledons, option B, root, option C, shoot apex, option D, plumule. Correct answer is option A, cotyledons. This is the heart-shaped stage formed due to the formation of cotyledons, which later form the cotyledon. The next question number 37. The two cotyledons arise from option A, embryonal axis, option B, hypocotyle, option C, epiblast, option D, plumule. Correct answer is option A, embryonal axis. The next question number 38. In dicot embryo, plumules lie between option A, cotyledon, option B, epicotyle, option C, hypocotyle, option D, radical. Correct answer is option A, cotyledon. This is the plumule which lie in between two cotyledon. These are the cotyledon. The plumule is lie in between cotyledons in dicot embryo. This is the plumule and these are the cotyledons. The next question number 39. The embryo becomes mature at option A, globular stage, option B, torpedo stage, option C, heart stage, option D, pro-embryo stage. Correct answer is option B, torpedo stage. The next question number 40. Crucifer type embryo is a typical of option A, Capsula bursa pastoris, option B, wheat, option C, maize, option D, rice. Correct answer is option A, Capsula bursa pastoris. The next question number 41. Heart-shaped embryo occur in option A, monocot, option B, dicot, option C, gymnosperm, option D, pteridophyte. Correct answer is option B, dicot. The next question number 42. In dicot embryo, the root apex is covered by option A, root cap, option B, hypocotyle, option C, suspensor, option D, radical. Correct answer is option A, root cap. The next question number 43. The embryonic shoot is called option A, plumule, option B, radical, option C, hypocotyle, option D, epicotyle. Correct answer is option A, plumule. The next question number 44. The embryonic root is option A, plumule, option B, radical, option C, cotyledon, option D, suspensor. Correct answer is option B, radical. The next question number 45. The terminal cell divide by option A, transverse division, option B, vertical division, option C, oblique division, option D, diagonal division. Correct answer is option B, vertical division. The next question number 46. The basal cell of suspensor enlarges to form option A, haustorium, option B, hypophysis, option C, endosperm, option D, cotyledon. Correct answer is option A, haustorium. The next question number 47. Embryo proper is derived from option A, terminal cell, option B, basal cell, option C, hypophysis, option D, suspensor. Correct answer is option A, terminal cell. The next question number 48. The embryo become heart-shaped due to option A, cotyledon formation, option B, root formation, option C, shoot apex elongation, option D, endosperm absorption. Correct answer is option A, cotyledon formation. The next question number 49. Torpedo-shaped embryo appear after option A, heart-shaped stage, option B, globular stage, option C, mature stage, option D, zygote stage. Correct answer is option A, heart-shaped stage. This is the heart-shaped stage. Then it form torpedo stage. The next question number 50. The terminal end of embryo form option A, plumule, option B, radical, option C, root cap, option D, suspensor. Correct answer is option A, plumule. The next question number 51. The basal end of embryo form option A root, option B cotyledon, option C plumule, option D endosperm. Correct answer is option A root. The next question number 52. The embryonal axis bear option A plumule and radical, option B two cotyledon, option C hypocotyl only, option D coliptyl. Correct answer is option A plumule and radical. The next question number 53. Dicots usually store food in option A cotyledons, option B endosperm, option C embryo sac, option D pericarp. Correct answer is option A cotyledons. The next question number 54. The first recognizable structure of a dicot embryo is option A globular embryo, option B heart embryo, option C torpedo embryo, option D suspensor. Correct answer is option A globular embryo. The next question number 55. In dicot embryo, endosperm is option A persistence, option B consumed, option C absent, option D partial. Correct answer is option B consumed. The next question number 56. Which stage in the development of embryo is almost similar in monocot and in dicot? Option A torpedo-shaped stage, option B heart-shaped stage, option C octant stage, option D globular stage. Correct answer is option C octant stage. The next question number 57. The division of zygote in the development of crucifer type of embryo is option A transverse, option B vertical, option C both and B, option D none of these. Correct answer is option A transverse. The next question number 58. In piperet type, the zygote divides by option A transverse division, option B vertical division, option C oblique division, option D random division. Correct answer is option B vertical division. The next question number 59. In which type of embryogeny or development of embryo the first division of zygote is oblique? Option A onagrade type, option B graminar type, option C solanare type, option D chenopodial type. Correct answer is option B graminar type. Example is triticum. The next question number 60. Crucifer type is also known as option A onagar type, option B astera type, option C solanar type, option D chenopodial type. Correct answer is option A onagar type. The next question number 61. In which type of embryogeny both apical and basal cell take part in development of embryo proper? Option A crucifer type, option B astera type, option C solanar type, option D onagar type. Correct answer is option B astera type. The next question number 62. In solanar type, the basal cell contribute to option A embryo proper, option B suspensor only, option C endosperm, option D cotyledon. Correct answer is option B suspensor only. The next question number 63. In chenopodial type, both basal and terminal cells, option A form embryo proper, option B form suspensor, option C are non-functional, option D form endosperm. Correct answer is option A form embryo proper. The next question number 64. Shoot or stem develops from option A plumule, option B radical, option C both and B, option D none of the above. Correct answer is option A plumule. The next question number 65. The radical elongate and form dash. Option A primary root, option B secondary root, option C rootlets, option D tertiary root. Correct answer is option A primary root. The next question number 66. The part of seed which grows and develop into shoot on germination is option A radical, option B plumule, option C cotyledon, option D ovule. Correct answer is option B plumule. The next question number 67. Future sporophytic generation in a seed is option A endosperm, option B perisperm, option C embryo, option D hilum. Correct answer is option C embryo. Dicot embryogeny or development of dicot embryo. The zygote cell first divide transversely to form upper cell and lower basal cell. The upper cell divide to form embryonal cell or embryo proper, which later divide to form proembryo and which again divide to form octant stage, which again divide to form two cotyledons and the shoot apex, that is the plumule, and plumule gives rise to shoot, and the lower part is from the radical and which later form the root. In the basal cell form the suspensor. 6 to 8 cells of suspensor are formed. The larger cell is known as the haustorium, and the middle part is the suspensor, and the upper part of suspensor is the hypophysis. From this hypophysis, radical are formed, and this radical is the apex part of the root. This is the development of monocot embryo. Here the zygote divide transversely to form terminal cell and basal cell. Again the terminal cell divide to form top cell and middle cell. The top cell divide to form plumule and single cotyledon, that is called scutellum, and the middle cell again divide to form hypocotyle and radical. In the basal cell form the suspensor cell, it is a single cell which functions as a haustorium. The next question number 68. Most common embryo development type in monocot is option A crucifer type, option B graminaceous type, option C solanite type, option D asteroid type. Correct answer is option B graminaceous type. The next question number 69. In monocots, the cotyledon is called option A scutellum, option B plumule, option C coleoptile, option D coleorhiza. Correct answer is option A scutellum. This is the cotyledon or single cotyledon which is called scutellum. The next question number 70. The embryonic shoot in monocot is enclosed by option A coleoptile, option B coleorhiza, option C scutellum, option D endosperm. Correct answer is option A coleoptile. The embryonic shoot in monocot is enclosed by coleoptile. The next question number 71. The embryonic root in monocot is enclosed by option A coleorhiza, option B coleoptile, option C scutellum, option D suspensor. Correct answer is option A coleorhiza. The next question number 72. The major food absorbing structure of monocot embryo is option A scutellum, option B endosperm, option C coleoptile, option D radical. Correct answer is option A scutellum. This is the coleoptile which cover the shoot apex, and this is the coleorhiza which cover the embryonic root. This is the epiblast, and this is the shoot apex, and this is the cotyledon or scutellum. The next question number 73. The first division of zygote in monocot is option A transverse, option B vertical, option C random, option D diagonal. Correct answer is option A transverse. The next question number 74. Embryogeny of monocot resembles dicot up to option A globular stage, option B heart stage, option C mature stage, option D torpedo stage. Correct answer is option A globular stage. The next question number 75. The scutellum is situated option A lateral to the embryonal axis, option B above plumule, option C below radical, option D on suspensor. Correct answer is option A lateral to the embryonal axis. This is the embryonal axis, and the scutellum is formed lateral to the embryonal axis. The next question number 76. The radical in monocot embryo is covered by option A coleorhiza, option B coleoptile, option C scutellum, option D pericarp. Correct answer is option A coleorhiza. The next question number 77. The plumule is protected by option A coleoptile, option B scutellum, option C coleorhiza, option D endosperm. Correct answer is option A coleoptile. The next question number 78. The zygote of monocot divide into option A 2 unequal cell, option B 2 equal cell, option C 4 equal cell, option D 1 suspensor cell. Correct answer is option A 2 unequal cell. The next question number 79. In monocot embryo, the cotyledon is option A terminal, option B lateral, option C basal, option D apical. Correct answer is option B lateral. The next question number 80. The part of embryonal axis below the level of scutellum is option A hypocotyle radical region, option B epicotyle, option C plumule, option D coleoptile. Correct answer is hypocotyle radical region. The next question number 81. The part above the level of scutellum is option A epicotyle, option B plumule, option C coleoptile, option D all of these. Correct answer is option D all of these. The part of embryonal axis below the level of scutellum is a hypocotyle radical region, and the part above the level of scutellum is a epicotyle, plumule, coleoptile. That is all of the above. This is the scutellum part. Above the level of scutellum is a coleoptile, plumule, and epicotyle. And below the level of scutellum is a radical and hypocotyle. This is the hypocotyle region and radical. This is. The next question number 82. The function of coleoptile is option A protecting plumule, option B protecting radical, option C absorbing food, option D producing endosperm. Correct answer is option A protecting plumule. The next question number 83. The function of coleorhiza is option A protecting radical, option B protecting plumule, option C absorbing food, option D storage. Correct answer is option A protecting radical. The next question number 84. In cereals, the scutellum absorbs food from option A endosperm, option B alluron layer, option C seed coat, option D suspensor. Correct answer is option A endosperm. The next question number 85. The embryonal axis in monocot lies between option A scutellum and coleorhiza, option B coleoptile and endosperm, option C cotyledon and radical, option D pericarp and seed coat. Correct answer is option A scutellum and coleorhiza. The next question number 86. The first cell of suspensor in monocot functions as option A hostodium, option B hypophysis, option C cotyledon, option D radical. Correct answer is option A hostodium. The next question number 87. The endosperm in monocot is option A persistence, option B consumed, option C absent, option D partial. Correct answer is option A persistence. The next question number 88. The coleoptile is a option A protective sheath, option B cotyledon, option C root cap, option D food organ. Correct answer is option A protective sheath which protect the shoot apex or plumule. The next question number 89. The embryo of monocot grain is option A small and lateral, option B large and terminal, option C central, option D peripheral. Correct answer is option A small and lateral. The next question number 90. In monocot embryo, option A scutellum is absent, option B 2 cotyledon are found, option C suspensor cell is a 6 to 10 layer, option D suspensor is single celled. Correct answer is option D suspensor is single celled. It is most important. This is the suspensor cell which is single celled in case of monocot, but in case of dicot it is 6 to 10 layer. The next question number 91. The embryo proper form option A plumule and radical, option B endosperm, option C coleoptile only, option D pericarp. Correct answer is option A plumule and radical. The next question number 92. Scutellum is a part of option A leaf bud, option B dicot embryo, option C monocot embryo, option D none of the above. Correct answer is option C monocot embryo. The next question number 93. Scutellum is a part of option A embryo, option B endosperm, option C seed coat, option D pericarp. Correct answer is option A embryo. The next question number 94. In monocot embryo, food is absorbed by option A scutellum, option B endosperm, option C alluron layer, option D pericarp. Correct answer is option A scutellum. The next question number 95. The scutellum is equivalent to option A cotyledon of dicot, option B plumule of dicot, option C endosperm, option D suspensor. Correct answer is option A cotyledon of dicot. The next question number 96. Scutellum is option A endosperm, option B cotyledon, option C alluron layer, option D embryo. Correct answer is option B cotyledon. The next question number 97. Scutellum is option A protective covering of the radical, option B a protective covering of the plumule, option C an endosperm, option D a seed-shaped cotyledon. Correct answer is option D a shield-shaped cotyledon. The next question number 98. Area between base of coleoptile and origin of scutellum is called Option A epicotyle, option B hypocotyle, option C mesocotyle, option D pseudocotyle. Correct answer is option C mesocotyle. The next question number 99. In a seed of maize, scutellum is considered as a cotyledon because it option A protect the embryo, option B contain food for the embryo, option C absorb food material and supply them to the embryo, option D convert itself into a monocotyl. Correct answer is option C absorb food material and supply them to the embryo. The next question number 100. Scutellum is option A food storing haploid structure in grass embryo, option B remnant of cotyledon in maize, option C shield shaped and large cotyledon of grasses, option D protective covering of plumule in grasses. Correct answer is option C shield shaped and large cotyledon of grasses. The next question number 101. Epiblast in monocot embryo is considered as option A second embryo, option B one-celled suspensor, option C nutritive tissue, option D rudimentary cotyledon. Correct answer is option D rudimentary cotyledon. The next question number 102. Epiblast present in certain monocot embryo represent option A rudimentary leaves, option B mesocotyle, option C scutellum, option D second cotyledon. Correct answer is option D second cotyledon. The next question number 103. Coleorrhiza and coleoptile are the structure associated with the embryo of option A monocot, option B dicot, option C angiosperm, option D spermatophyte. Correct answer is option A monocot. The next question number 104. Coleoptile and coleorrhiza in monocot embryo. Option A are extension of scutellum. Option B are foliar structure. Option C are extension of plumulin radicle. Option D secret hydrolytic enzyme. Correct answer is option A are extension of scutellum. The next question number 105. In monocot embryo, the radicle is produced by option A terminal cell, option B middle cell, option C epiblast, option D suspensor. Correct answer is option B middle cell. It is most important. The radicle is produced by middle cells. The next question number 106. In a monocot embryo, the radicle and root cap are enclosed in option A plumule, option B scutellum, option C coleoptile, option D coleorrhiza. Correct answer is option D coleorrhiza. The next question number 107. The portion of the embryonal axis above the level of attachment of scutellum is option A hypocotyle, option B epicotyle, option C radical, option D coleorrhiza. Correct answer is option B coleoptile. The next question number 108. Epiblast found in the seed of wheat and rice is an outgrowth of option A opposite origin of scutellum, option B at the tip of coleorrhiza, option C midway on coleoptile, option D at the tip of radical. Correct answer is option A opposite origin of scutellum. The next question number 109. In some seeds like black pepper and beet, remnant of nucellus are present. What is this residual persistence nucellus called? Option A alluron layer, option B endothelium, option C perisperm, option D epiblast. Correct answer is option C perisperm. It is most important. The next question number 110. Alluron layer take part in option A protein of delicate embryo, option B enzyme synthesis, option C transfer of food to cotyledon, option D transfer of food from cotyledon to embryo tip. Correct answer is option B enzyme synthesis. The next question number 111. Alluron layer is option A outer layer of scutellum in contact with endosperm, option B layer of pericarp specialized in absorption of water, option C layer present in the ovule that guide pollen tube, option D layer present on the outside of endosperm and having protein grain. Correct answer is option D layer present on the outside of endosperm and having protein grains. The next question number 112. The alluron layer is present in option A dicot seed, option B monocot seed, monocot endospermic seed, option C non-endospermic seed, option D embryo only. Correct answer is option B monocot endospermic seed. The next question number 113. The alluron layer is single-celled and located option A between pericarp and testa, option B between endosperm and seed coat, option C around endosperm inside the seed coat, option D in embryo axis. Correct answer is option C around endosperm inside the seed coat. The next question number 114. In maize, alluron layer develop in outermost region of option A pericarp, option B epicarp, option C testa, option D endosperm. Correct answer is option D endosperm. This is the endosperm part and this is the alluron layer. And this alluron layer is the outermost part of endosperm. Here is also this is endosperm and this is the alluron layer and this is the seed coat. The next question number 115. Alluron layer is rich in option A protein, option B starch, option C lipid, option D oxygen. Correct answer is option A protein. The next question number 116. Alluron layer helps in option A storage of food in endosperm, option B protection of embryo, option C utilization of stored food, option D all of the above. Correct answer is option C utilization of stored food. The next question number 117. This is the outer covering of endosperm which separate the embryo in monocotyledon seed. Option A seed coat, option B coleoptile, option C coleorrhea, option D alluron layer. Correct answer is option D alluron layer. The next question number 118. In maize seed, the outer covering of endosperm separate the embryo by a proteinaceous layer called option A coleoptile, option B alluron layer, option C scutellum, option D coleorrhea. Correct answer is option B alluron layer. The next question number 119. Alluron layer is a part of option A endosperm, option B embryo, option C tegmen, option D testa. Correct answer is option A endosperm. The next question number 120. At the onset of seed germination, the digestive enzyme amylase are produced by the action of or the activity of alpha amylase in the endosperm of a germinating seed of barley is induced by option A ethylene, option B cytokinin, option C indolacetic acid, option D gibberellin. Correct answer is option D gibberellin. The next question number 121. An enzyme that can stimulate germination of a barley seed is option A protease, option B invertase, option C alpha amylase, option D lipase. Correct answer is option C alpha amylase. The next question number 122. The main function of alluron layer is option A photosynthesis, option B secreting of hydrolytic enzymes, option C protection, option D food storage. Correct answer is option B secretion of hydrolytic enzymes. The next question number 123. The alluron layer secret option A amylase, option B protease, option C lipase, option D all of these. Correct answer is option D all of these. The alluron layer secret various hydrolytic enzyme during seed germination. The next question number 124. Which hormone activate the alluron layer during seed germination? Option A auxin, option B gibberellin, option C cytokinin, option D abscisic acid. Correct answer is option B gibberellins. The next question number 125. The enzyme secreted by alluron layer helps in option A synthesizing starch, option B breaking down stored food in endosperm, option C photosynthesis, option D root elongation. Correct answer is option B breaking down stored food in endosperm. The next question number 126. In maize grain, the alluron layer lies option A outside the endosperm, option B inside the endosperm, option C between the pericarp and endosperm, option D in embryo. Correct answer is option C between pericarp and endosperm. The next question number 127. The alluron layer mainly store option A protein granule, option B starch grain, option C lipid body, option D calcium oxalate crystals. Correct answer is option A protein granule. The next question number 128. The alluron layer converts stored food or stored starch of endosperm into option A glucose and maltose, option B protein, option C fat, option D lipid. Correct answer is option A glucose and maltose. It converts starch into glucose and maltose. The next question number 129. The alluron layer disappears option A during seed development, option B during seed germination, option C after fertilization, option D before endosperm formation. Correct answer is option B during seed germination. The next question number 130. The cells of alluron layer are Option A, dead and lignified. Option B, living and protein rich. Option C, meristematic. Option D, non-living. Correct answer is option B, living and protein rich. The next question number 131. Suspense are formed during embryogeny of Sagittaria is option A 6 to 10 celled, option B 4 to 5 celled, option C 1 celled, option D 2 to 3 celled. Correct answer is option C 1 celled. It is a genus, it is a aquatic plant. The next question number 132. The first division of zygote is oblique in option A solanum, option B aster, option C triticum, option D najas. Correct answer is option C triticum. The next question number 133. Perisperm is option A persistence or remnant of nucellus, option B degenerated secondary nucleus, option C degenerated synergid in seed, option D remain of endosperm. Correct answer is option A persistence or remnant of nucellus. The next question number 134. Perisperm differ from endosperm in option A being a diploid tissue, option B its formation by fusion of secondary nucleus with several sperm, option C being a haploid tissue, option D having no reserve foot. Correct answer is option A being a diploid tissue. Perisperm is diploid whereas endosperm is triploid. The next question number 135. Peri Perispermic seeds are option A castor, sunflower, option B black pepper, beet, option C maize, beet, option D barley, maize. Correct answer is option B black pepper, beet. The next question number 136. An example of a seed with endosperm, perisperm and carancule is option A lily, option B castor, option C cotton, option D coffee. Correct answer is option B castor. The scientific name of castor seed is Ricinus communis. The next question number 137. What is the haploid of perisperm? Option A haploid, option B diploid, option C triploid, option D tetraploid. Correct answer is option B diploid. This is the first cell of a suspensor which is large. It is called haustorium or vesicular cell, and this is the last cell of a suspensor which is called hypophysis. It forms the radical tip, and the haustorium or the vesicles are present at the micropylar end, and the embryogenal mass are present at the chalazal end. Please like, comment, share and subscribe my channel. Thank you for watching this video.